Thymosin Beta-4 versus TB-500. Today, we're cutting straight through the noise. We are separating the full-length, naturally occurring protein from the heavily marketed synthetic fragment. You see, there's a lot of confusion out there, so we need to look at the exact, hard biological reality of these two very different molecules. Here is our roadmap. We're defining thymosin beta-4, looking at the TB-500 synthetic fragment, and examining human clinical trial evidence. Then, we'll cover what TB-500 is biologically missing, the recent Rakerman metabolite finding, and finally, pharmaceutical development choices.
Defining thymosin beta-4, the biological baseline.
So, thymosin beta-4 is this 43 amino acid protein, first isolated way back in 1966 from thymic tissue. And the crazy thing is, it's found in virtually every nucleated human cell. It's actually encoded directly on your X chromosome. When you get hurt, your body naturally rushes this protein right to the site of healing, so it gets highly concentrated in your platelets, white blood cells, and wound fluids. Plus, it has this central actin binding region right between positions 17 and 23 that is absolutely crucial for helping cells move. But it's not just about movement. T-beta-4 also regulates some pretty vital repair pathways involving a protein called ILK, or integrin-linked kinase. ILK basically helps cells sense and respond to their surroundings. Think of it like a master signaling hub, sitting right there where cells attach to the extracellular matrix. And full-length T-beta-4 is what actively engages this hub. When T-beta-4 activates ILK, it sets off this highly specific cascade of beneficial actions. First, it helps cells migrate toward damaged tissue. Second, it promotes cell survival right after an injury. Third, it supports the formation of brand new blood vessels. And fourth, it actually switches on dormant repair cells to help heal the heart. Now, this isn't just theory. The foundational evidence here comes from a landmark 2004 study by Bach, Marquette, and colleagues. They demonstrated that T-beta-4 activates this exact ILK-dependent pathway, which is literally required for cardiac regeneration. Then we have PINCH. This is an adapter or scaffold protein. It binds to ILK and another protein called parvin to form the IPP complex. This whole complex connects cells to the extracellular matrix, stabilizes their internal skeleton, and coordinates their movement during tissue repair. Now, the exact molecular relationship here is a bit less established than with ILK, but studies do report that T-beta-4 influences this pathway too, promoting cell survival. So the absolutely crucial point to remember here is that the full T-beta-4 protein utilizes a massive range of biological functions across its entire 43 amino acid structure.
The TB-500 synthetic fragment, a synthetic peptide.
Okay. The physical difference between these two is just immense. As we said, full-length thymosin beta-4 has a 43 amino acid structure, weighing nearly 5,000 Daltons. TB-500, on the other hand, is just a synthetic fragment. It contains only a 7 amino acid sequence, taken right from positions 17 to 23 of the full protein. It's protected by a small chemical modification and weighs exactly 889 Daltons. It isolates only that central actin binding region we talked about earlier, making it roughly 6 times lighter than the real deal. Now, the logic behind creating this was to isolate the specific part responsible for healing and make a stable, easy-to-produce peptide. But here is exactly why that matters. Because TB-500 is so much smaller, it absorbs, distributes, and breaks down entirely differently in your body. And because it's just a tiny fraction of the full protein, it might not actually reproduce all the biological effects we see with full-length T-beta-4.
Human clinical trial evidence, examining the data.
I want to read this quote directly from the ONPEPS analysis because we need absolute clarity on this. It says, "Peptide practitioners and vendors who are citing human clinical evidence for TB-500 are mostly taking their data from T-beta-4 studies. TB-500 has not been tested in human clinical trials." You guys, this is a massive distinction. Yes, the foundational wound healing work by Philp and colleagues back in 2003 used the 7 amino acid actin binding fragment in diabetic and aged mice. But TB-500 itself has absolutely 0 human clinical trials of its own. None.
Whenever human clinical evidence is cited in marketing, specific trials get referenced. Take the SEER-1 Phase 3 trial for a rare eye disease. They used RGN-259, which is a formulation of full-length T-beta-4. The ARISE-3 trial for dry eye disease, full-length T-beta-4. The 2016 JU cardiac pilot study used intravenous T-beta-4, and the Phase 2 trial for venous stasis ulcers used topical T-beta-4. The data objectively demonstrates that these human trials rely on the full-length protein, not the TB-500 fragment.
What TB-500 is missing? Biological limitations. So what are the potential missing functions? First up is heart repair. TB-500 completely lacks the C-terminal region of the full protein, and that specific region is exactly what activates ILK to move cells, support new blood vessels, and wake up dormant heart repair cells. Without it, TB-500 may not activate ILK to the same extent. Second is anti-scarring effects. There was a fascinating 2025 study by Marr and colleagues finding that full T-beta-4 helps prevent scar tissue by shutting down a protein called ROC1. That protein stops regular fibroblasts from turning into scar tissue-creating myofibroblasts. But researchers haven't identified which part of T-beta-4 does this, so we literally have no idea if the tiny TB-500 fragment retains this ability. Finally, cell survival. It remains totally unknown if TB-500 can influence the ILK pinch signaling pathway. Now, I'm not saying TB-500 is inactive. Remember, that mouse study did show wound healing activity for the fragment itself. It just means the full-length protein has a much broader functional range that the fragment is unlikely to fully reproduce.
The Rahman metabolite finding, a 2024 complication.
This finding is from 2024. It was published in the Journal of Chromatography B by Rahman and a team of researchers. The sheer recency of this finding highlights just how actively this area is still being investigated today, and it honestly challenges a lot of the standard narrative you'll find online. They looked at how TB-500 breaks down in the body, and what they found is that TB-500 converts into an even smaller metabolite called ACK-LKKTE. The resulting hypothesis is pretty profound. All those wound-healing benefits people credit to TB-500, they might actually be coming from this smaller metabolite, not TB-500 itself. If that's true, TB-500 might simply be acting as a delivery vehicle, something that just converts into the molecule that actually does the physical work. It would still explain the observed effects, for sure, but it adds another really significant wrinkle to the biology. However, we have to preserve a healthy dose of scientific hesitation here. This finding remains unconfirmed by independent researchers as of mid-2026. While it has shown up in some later papers, it lacks independent verification. Grasping this nuance is absolutely critical if we want a complete objective understanding of the current research landscape. Pharmaceutical development choices, bridging the gap. The reality here is pretty stark. No pharmaceutical company has pursued the TB-500 fragment as a drug candidate for any indication whatsoever. Period. Companies like Regenerex deliberately chose to use full-length T-beta-4 throughout their clinical development, and that was a deliberate choice, reflecting exactly what we've been discussing. The broader functional profile of the intact protein triggers a much wider range of systemic healing and cell survival pathways that the tiny fragment simply cannot guarantee it replicates. This leads us to a highly necessary conclusion about how to read the science. Clinical evidence for Thymosin Beta-4 and clinical evidence for TB-500 are not interchangeable terms. When a study reports that TB-4 accelerated wound healing or reduced cardiac fibrosis or promoted corneal repair, you cannot just directly transfer that finding to injectable TB-500. Yes, the mechanism clearly overlaps in the actin-binding domain, but the full protein has functional domains the fragment completely lacks. Vendors constantly conflate the two, using the full protein's human data to market the fragment, but the science demands we keep them separate. Which brings us to a final provocative thought. If the active molecule is ultimately proven to be the smaller ACK-LKKT metabolite, how much of the current consumer knowledge about TB-500 needs to be fundamentally rewritten? The immense complexity of peptide research is still unfolding right in front of us. What we know for sure is that relying on conflated marketing claims ignores the biological realities. We have to continue questioning what exactly is doing the work at the cellular level.